I'm going to live, and in workflow, you have to continue within 1 minute. So, Mr. Yahya, Mr. Workflow will invite you officially, and then we can start. Thank you so much.
Oh, thank you. I'm grateful.
Assalamu alaikum and a very warm welcome to everyone. On behalf of VitaNova International Alliance for Sciences, I am delighted to welcome all of you to the Advancements in Molecular Biology course 2026. I am Nupu Akhtar, the head of collaborations of VNIAS, your host for today's session, and it is a pleasure to have you with us as we begin this exciting academic journey. Today's topic is omics technologies in modern biotechnology. Omics technologies have transformed biological research by enabling scientists to study genes, proteins, and metabolics and other biological systems on a large scale. These approaches play a vital role in precision medicine and drug discovery, agriculture, as well as biotechnology. Now, it is my honor to introduce our distinguished instructor, Mr. Yahya Yunusa Riko, founder of the Research and Molecular Genetics Center for Health, Chiang University, Thailand. This expertise in molecular biology and genetics has inspired many inspiring researchers, and we are truly grateful to have him with us today. Without, without further delay, I would like to invite Mr. Yahya Yunus Rico to begin today's session. Uh, the virtual floor is yours now.
All right. Thank you very much for your kind words of welcome and introduction, everybody. Greetings of the day and assalamu alaikum. I hope I am audible.
Yes, you are audible. You can proceed with the session.
Okay, thank you very much. Please, we can move to the next slide. I'm here to talk about omics technologies in modern biotechnology. Obviously, looking at the outline of the slides, we have a lot to cover. I would try to make the presentation short and easy to follow. And in case anybody has any comments or questions, we can ask them at the end, or you can reach out to me personally at the end of the session. So what we are covering in today's presentation include the following: a brief introduction about myself and my role in biotechnology research. Even though I'm just starting, I'm an early career researcher, and I consider it a privilege to be allowed this platform to speak, where my mentors like Professor YY Dini are also speakers. I really consider this a great honor, and I appreciate VitaNova International Alliance for Sciences for giving me this opportunity. Then, as usual, we start with a story to illustrate the theme of the lecture, and then we move on to etymology of multi-omics and biotechnology, then the objectives of the presentation, and then we differentiate between oms and omics, and then we enter into multi-omics, and then the central dogma, which is about genomics, transcriptomics, and proteomics. Then we look at the pipeline for all of these studies, and we look at some hypothetical data, and then we look at the relevance and application of omics in modern biotechnology. Then we summarize the lecture, we do one little evaluation task, and references and acknowledgement. We can move to the next slide. Thank you. All right, so I am, as I said, an early career microbiologist who is currently studying for his PhD at Chiang University, Thailand. My area of focus in research includes omics studies, especially plant microbe interactions, and also One Health, which is how the health of humans and animals is connected to the environment. I am fortunate to have collaborations with different researchers in various fields. And my PhD work is currently about stress tolerance in plants, especially from the multi-omics angle. We can move to the next slide. Thank you. So if somebody asks me, what are the three most important areas today in terms of life sciences research, I would say they are bioinformatics, biotechnology, and multi-omics. And the reason I say this is if you look at all the fields, agriculture, ecology, public health, system biology, they all revolve around these three key areas. And in this session today, we'll try and cover biotechnology and multi-omics. Bioinformatics would be taken care of by other speakers subsequently. So we start with a story. As a teacher, I like to begin my lectures with a story to illustrate the point I am driving home. This story is about the blind man and the elephant. It's a very popular story that some blind men were asked to identify an elephant, but by touching different parts of the elephant. So one of them touched the trunk of the elephant and he said, okay, this is a snake. Another person touched the ear and he said, this is a fan. Another person touched the trunk and he said, this is a spear. Another person touched the leg. He said, this is a tree. And yet again, another blind man touched the side and he said, this is a wall. Another one touched the tail and he said, this is a rock. So the idea here is all the blind men are touching different parts of the elephant and they are identifying it wrongly. And the reason they are identifying it wrongly is they do not have the full picture of the elephant. They are only having partial pictures of the elephant. So the link of this to multi-omics is if, as a scientist, you are studying genomics alone, or proteomics alone, or transcriptomics alone, you are only studying the DNA or the RNA or the protein, you are not getting the whole picture. If you want to get the whole picture, you need to look at the elephant as a whole, then you can identify, okay, this is an elephant. That's why we have this quote from René Dubois, one French ecologist. He said, the only thing worse than being unable to see the forest for the trees is being unable to see the trees for the forest. We are in slide number 4. Thank you. So the meaning of this quote by René Dubois is, sometimes people get lost in the details and they fail to see the big picture. Somebody would be talking about a gene without relating that gene to the RNA transcript, without relating that RNA transcript to the protein. So this is wrong. And sometimes we are unable to see the trees for the forest. Somebody would be looking at the big picture. Okay, bacteria improve plant growth, but how? Without going into the details. So both of the approaches are wrong. So we need to look at the individual parts of the elephant and also the elephant as a whole. So in modern biotechnology, what we want to do is we want to merge the layers, the genomics, the transcriptomics, the proteomics. Only then can we get the whole picture. We can move to the next slide. Thank you. So etymologically speaking, what is the meaning of the word omics? So the word omics is from the word om, and om means the complete set or totality of something. So if you say, for example, genomics, you mean the complete or total of the genome. If you say proteomics, the complete total protein that is expressed by an organism. Transcriptomics, the RNA transcripts, like that. So if you add X to it, it becomes the field of study. So proteome, for example, is the complete set of protein that is expressed by an organism. And then proteomics is the study of the complete set of protein that is expressed by an organism. So we take omics and we apply it in biotechnology. I think the word biotechnology was first used around 1919, while multi-omics was first used around 2012, I think. So biotechnology is from the word bio, which means life, and technology, which is the application of scientific knowledge to solve human problems or to improve life. So we're applying our knowledge of omics to solve human problems. Let's look down memory lane. How have we progressed? We have come a long way from Aristotle describing living things, Carolus Linnaeus classifying living things, Van Leeuwenhoek, Antoine van Leeuwenhoek, who first observed microorganisms microscopically, Gregor Johannes Mendel, who talked about genetics, Louis Pasteur, who laid the foundations of microbiology, Watson and Crick, who described the structure of the DNA, recombinant DNA sequencing around the 70s, PCR around the 90s, then the Human Genome Project at the turn of the 21st century. Later, we are now talking about systems biology and multi-omics. So if you look at it, we are moving from the organism level down to the protein, metabolome, and genes level. And let us take home this quote: no single layer explains life. Life is too complex to be explained by a single gene or an RNA transcript or a single protein. What we need is we need to integrate the knowledge from all three aspects for us to get insight. So the difference between traditional biotechnology, because we said multi-omics in modern biotechnology, and modern biotechnology is that traditional biotechnology is talking about like fermentation, like breeding, uncontrolled and guided biological processes. But modern biotechnology is based on precision, genetic engineering, modification of organisms or their genes through CRISPR and other recombinant DNA technology. Next slide, please. Thank you. So I want us to achieve three things in this session. Number one, for us to explain the fundamentals of omics technologies. And number two is to design and apply this knowledge in our experiments. And then number three is to interpret the data. Because some people make the mistake of entering into omics, genomics, proteomics, transcriptomics without having an understanding. That is why even in practicals, before you start, you do the pre-lab. They explain to you the concepts, because grasping the concepts is very important. If you start the practical without knowing the concepts, what happens if you get a problem? Then you go back to the theory. So we start with the theory first, and then we look at how the experiments are designed. And then we look at, if you have the data, how can you make sure you identify the elephant and don't say it's a snake or it's a wall or something. And then how do you make sure that you see both the forest, which is the large picture, and the trees, which is the individual detail. So three things: explain omics, design your own experiments, and interpret the data. Next slide. Thank you. So the ohms, there are so many. I only captured some here. And the slide looks chunked up. I'm sorry for that, because there are a lot of things there. So genome is talking about the whole gene. Exome is talking about exon. You remember the exon and the intron. The exon is the coding part. So exome is talking about the coding part of the genome. We have epigenome, epigenetics. You remember the regulation of DNA expression, epigenetics. So that's the epigenome. Transcriptome is talking about the RNA. Proteome is talking about protein. Lipidome is talking about lipid interactions, especially at the membrane level. Ionome is talking about ions, all the metals and sulfur and phosphorus and how they interact with the cell or regular cellular processes. Metalome is talking specifically about metals. While to differentiate it from ionome is all the ions. Remember, ions can be metals or non-metals, but metalome is specifically talking about metals. Phenome, which is the observable trait. Remember phenotype and genotype. So phenome is the observable trait. In bacteria, for example, what are the biochemical tests, properties that you can see on the bacteria? Virome is talking about viruses. Microbiome, microorganisms as a whole. Mycobiome, fungi. Rhizobiome, the microorganisms that are associated with the roots, rhizosphere, like that. And each of these forms, if you add X to it, it becomes the study. So for example, genomics is the study of the genome. Lipidomics is the study of the lipidome. Microbiomics is the study of the microbiome, like that. So the omics are the biological layers. The ohms are the biological layers, while the omics are the methods of studying them. We have a diagram of a cell. I just illustrating what I just finished explaining. We have the transcriptome, which is the mRNA products. We have the metabolome, which are the small molecules, metabolites that are byproducts of cellular metabolism. We study them under metabolomics. Next slide, please. Thank you. So how do we combine omics? Because remember from the story I told you at the beginning, we can't gain insight by just looking at a single omic. We need to combine them. So if we combine omic studies from different environments or different communities or different samples, then we have what is called multi-omics. So for example, in a community, you can have the virome, which is the total viral component of the community. You have the bacteriome, which is the total bacterial component of the community. You have the microbiome, which is the fungal. So if you combine a study of all of this, then you are studying the microbiome. Then you can combine it with, for example, proteomics. You are studying the protein. You can combine it with transcriptomics. You are studying the transcriptome. So if you have this combined data set, then we say you are doing multi-omics study. And I want to introduce something here, meta-omics. Meta-omics is talking about specific environments. So for example, when we say metagenome, we are talking about the genome in specific environments. For example, crude oil contaminated soils. If you're studying the genes that are expressed in crude oil contaminated soils, something like ALKB gene, alkane hydroxylase gene. So what you are doing here is you are doing a metagenomic study. You are studying the genome, but in a specific environment. Somebody who is in animal science, for instance, he is studying the gut microbiome of, say, ruminants. So what he's also doing there is a metagenomic study. So the meta-omics usually involve extracting and processing large data sets of DNA from a specified environment. And then you have multiple layers of that data set. You combine it together and perform community level analysis, and then you interpret the data set. Let me make one thing clear here. Meta-omics does not include metabolomics. Somebody can be confused because they said metabolomics. Metabolomics is different from the meta-omics. Meta-omics are like the metagenomics, meta-proteomics, but not metabolomic. That meta in metabolomics is referring to metabolism, not the meta of community. So multi-omics means combining. For example, typically people used to combine 4 things: metagenomics to study the genes, metatranscriptomics to study the RNA transcript, metaproteomics to study the proteins, and then metabolomics to study the metabolites, all in a single community if we are talking about the meta-analysis. You can also just say genomics, transcriptomics, proteomics, metabolomics, like that. So the key idea here is if you are integrating, then you are going to have the whole picture. We are going to see this later. Can we move to the next slide, please? Thank you. So let's look at the central dogma of modern biology. They said information flow is from the DNA to the RNA to the protein. The information is encoded in the DNA, and then it is transcribed into RNA, and then RNA is translated into protein. Now, at each of this level, some processes are occurring. From DNA to RNA is called transcription. From RNA to protein is called translation. And this sequence of events can happen backwards. So for example, RNA can be reverse transcribed into DNA, which is where the issue of the reverse transcription happens, like we see in HIV and other RNA viruses. And protein can also be used as a template to synthesize DNA. Even though here in the slide you can see they say no non-sequence information flow from protein back to nucleic acid. This is what you see in the textbooks. But recently, just 3 months ago, a paper was published in the journal Science. I give a citation for the paper in the slide. So Deng et al. 2026. They found out that there is a bacterial defense system in Escherichia coli, to be precise, that uses a reverse transcriptase enzyme to synthesize DNA. But there are a lot of caveats about the process. They are not saying that protein has been used to synthesize DNA. They're just demonstrating the potential and what have you. And I think the basic idea is simple to understand. Proteins are made of amino acids, and the amino acids are from RNA. So if you have a chain of amino acids, you can use that chain to decode the likely mRNA transcript that produces the amino acid that is used in the formation of the protein. And from that mRNA, you can easily decode the gene template that is used to synthesize the RNA. So this is part one of the genomics and transcriptomics and proteomics. So if we are studying at the DNA level, then we are doing genomics. If we are studying at the RNA level, then we are doing transcriptomics. And if we are studying at the protein level, then we are doing proteomics. Can we move to the next slide? Thank you. So let's look at it in more detail. So at the genomics level, which is where we start, we know that information is encoded in DNA. This information contains a lot of things. We have what is called the coding region of the DNA that is responsible for transcription and gene expression. And then we have the non-coding region. We have single nucleotide polymorphisms. We have genome-wise association studies. These are related to diseases like cancer, like schizophrenia. You know, DNA has fidelity. If there is an error in just a single base of the DNA, it can influence the causation of disease and a lot of other things there. So usually the techniques that we employ in genomics include PCR, polymerase chain reaction, for amplifying the DNA sequence. We have whole genome sequencing for sequencing the DNA, which is WGS. We have amplicon sequencing. Usually we use amplicon sequencing in communities. You remember the example I gave in animal science, somebody trying to study the community in the gut of the rumen. So they are going to use amplicon sequencing. And then they have this large voluminous data set that they analyze. We're going to see this later. So at the transcriptomics level, we are studying the RNA. And if we are studying the RNA, we are looking at the expression. Remember, we said it's not all part of the gene that is expressed into RNA. I think the percentage of bacterial DNA that is expressed into RNA is around 85 to 90%, something like that. So some part of the RNA is, of the DNA rather, is not expressed. It is made up of things like promoter sequences and other stuff, things like the epigenomics we talked about before, which are basically regulating the gene expression. So the part of the DNA that is transcribed into RNA is telling you what the cell is actually doing. It is telling you, okay, this cell is deciding to transcribe this part of its genome into this mRNA for further usage. And another important thing to note is that it is not all the transcribed RNA that is further translated into protein. And this is just linking to the idea that studying a single part like the genomics without integrating it is going to give you only a partial picture of the whole story. So in RNA transcriptomics, usually what we are doing is we are studying the amount of the gene expression. We can quantify it. Sometimes we use reverse transcriptase qPCR, quantitative polymerase chain reaction, to tell us, okay, this particular gene is being expressed as this level compared to this gene. That's why we have what is called differential gene expression. And we can say, okay, this gene, it's expressed at a higher level compared to this. And this is really applicable in diagnosis of disease. Proteomics, the final layer. So after the messenger RNA, and a very surprising thing is, it's only about 5% of the transcribed RNA that is translated into protein. So when the RNA is finally translated into protein, we know that the cell is ready for business. So we analyze the protein sequence using various techniques. We visualize its structure, and then we go into a field called Interactomics, which is talking about protein-protein interactions, which also have implications for disease. There are different methods that I use to study protein. We are going to see this later in the slide. So can we move to the next slide, please? Thank you. So let's start with the genomics. The slide is chalked up. I'm sorry. There are a lot of things. So I think I would give us a general overview. The session recordings will be available. We can come back and explore in more detail. So generally, if you're doing genomics, what you do is you start with the sample collection, and then you do the DNA extraction from your sample, and then you quantify the extracted DNA, and you do quality check. This is very important. in DNA or any multi-omics study that you know, the number one thing you should avoid is contamination. And remember, DNA is present in every life form. So even in your body, there is DNA that can contaminate the whole pipeline. So you have to be really careful about this. And then after you you extract rather the DNA. Sorry, I'm a microbiologist, so I mean it's the isolation of microorganisms. So if you extract the DNA, then you quantify it. You look at the quality, you check the absorbance at 260, 280, you make sure. Sometimes you use nanopore, a lot of techniques anyway. And after that, you move to the sequencing. For bacteria, we usually use 16S ribosomal DNA sequence. And the reason we use 16S is that the 16S gene is highly conserved across bacterial species, but it has some hypervariable regions, V1, V3, V4. These regions, they are highly mutable. They differ between bacterial species. So if you can sequence that region, you can use it to identify a bacterium. Whole genome sequencing. Whole genome sequencing is talking about trying to sequence the entire genome of an organism. It would give you the best detailed form of information that you can have about the organism, the coding sequences, the non-coding sequences, the GC content. We're going to see that later in the slide. And then what next? PCR, polymerase chain reaction. Because DNA quantity is very low, sometimes you find it in nanogram, things like that. So if you do polymerase chain reaction, you amplify that DNA millions of times to the extent that it becomes detectable. And if you have that detectable amount of the DNA, you can use it for downstream analysis, or maybe whole genome sequencing using Illumina and PacBio. There are a lot of platforms, and these platforms, they differ whether the expected target DNA to be sequenced is short or large, it depends. And then you have your output usually as FastQ, FastQ or faster. FastQ has quality scores attached to it, as opposed to faster, which is just the raw sequence. And then after you have obtained your data set, we move to the bioinformatics workflow. So usually you have a sequence of ASV. ASV means amplicon sequence variant. And then you go ahead and do taxonomic analysis. You do the phylogenetic tree, and you obtain alpha diversity. And okay, alpha diversity means like the number of organisms that you are targeting. So for instance, you are studying a bacterial community, and then you have Escherichia as a genus, you have Salmonella as a genus, you have Proteus as a genus. Let's assume it's the intestinal community you are studying. So the alpha diversity is at its basic level, the number of organisms that you have, different organisms. So here we have 3 organisms, Escherichia, Salmonella, and Proteus. So you say the alpha diversity is 3. This is obviously simplified. There are a lot of things involved, but I'm trying to keep everything simple here. Next is if you're doing whole genome sequencing, it's a bit more complex than normal 16S ribosomal sequencing. You need to do what is called annotation. The raw data that they are going to give you after the sequencing is unintelligible. You cannot understand it. But if you go to databases, there are databases dedicated for genome annotation study. They would annotate the genome for you and give you this concentric cycles. We're going to see that later, that are explained in the actual composition of the genome. So how do you interpret this? The first interpretation you do is you identify the organism. You blast and you identify it from NCBI database, and then you move to statistics, especially if you're doing community analysis. There are dedicated sequences that you analyze, that you can compare with. There are actually workflows developed in R, the R software for statistical computing, that you can use to help you analyze your data set. And usually we use the PermaNova. PermaNova is a bit different from the normal ANOVA we know. PermaNova means permutational analysis of variance. It's usually suited for microbial community data. And the reason is simple. Microbial community data is not normally distributed. You see a lot of zeros in it. So sometimes the distribution tends to be extremely clustered, maybe to the right or to the left, things like that. To the right usually. So if you use PermaNova, the analysis would be better. and then you go ahead and deposit your sequences in NCBI or ENA. ENA means the European Nucleotide Archive, and then you publish your results for other scientists to compare yours with this. There are some best practices and sources of error that we should avoid. I think these ones are easy to read and understand. We can, okay, one final thing before we move. In virology, the study of viruses, viruses are difficult. You don't use 16S ribosomal DNA analysis to study viruses because viruses are highly mutable. They rapidly mutate. We see that during COVID-19, how we are having different variants and what have you. So usually we use other specific sequencing platforms that their main aim is to eliminate contamination, because viral genomes can be contaminated by the host. So you'd be sequencing, you'd be trying to sequence a viral genome and you end up sequencing the host genome or something. So it's difficult to do. Next slide. Okay, transcriptomics. Here we are. So now we are looking at the RNA transcripts. Okay, we have seen the genome. You know, the genome is like telling you the total capability of the cell. This cell is capable of expressing 1,000, 10,000 genes. But the transcriptomics would tell you the actual number of genes that are being expressed. Okay, out of that 1,000 genes, the cell is currently expressing 500 genes. Why would the cell express 500 genes? Because of differences in physiology and metabolism. Cells are highly economical. They don't express genes randomly. Gene expression is tightly controlled because it's an energy-intensive process. So the cell would balance between its requirements and what needs to be expressed. Gene expression is usually not wasteful. So how do we do it? We also start with the extraction after sample collection, but here there are some caveats. RNA is unstable rather. So if you're not careful, you lose the RNA that you're trying to extract even from the sample collection stage. That's why usually we store it in liquid nitrogen or in other cold storage, and they tell you that you extract and sequence immediately to avoid the sample from degrading, things like that. So you also extract and you do the whole process like we did in genomics. You measure the quality and what have you, and then you go ahead and do sequencing. Sometimes they do reverse transcriptase qPCR. I think I mentioned this before, to sequence the RNA. And then you have the raw data. RNA raw data is more complex than DNA. You have a huge amount of data that the first thing that you need to do is try and trim the data and have RNA counts. Okay, you have a table. These are genes. I think we have a table subsequently in the slide that is mimicking the actual data set. These are the number of genes. These are their expression levels. And then another beautiful thing about transcriptomics is you can link the gene expression to pathways, specific pathways, because we know that genes are associated with specific pathways. Okay, this gene is potentially involved. I think this would be beautifully captured in biodegradation. This is something that I have a little expertise in. In biodegradation, if you have the particular genes that are being expressed, then you can putatively guess the pathway. Okay, based on this gene, which is converting this substrate to this product, we think the microorganism might be following this particular pathway to break down this substrate and produce this. And this has a reliable measure of accuracy to some extent. And another thing about transcriptomics is you have what is called log-FC. Log-FC means logarithmic fold change. Okay, this gene is being expressed twice compared to this gene. You know, gene expression can involve upregulation or downregulation. Okay, we have a baseline level. Usually the genes are being expressed, let's say 10 times a second. Now, in a particular condition, let's say environmental stress, we see that one gene has been reduced to 5 times a second. It means it is down-regulated. Another gene has been upregulated to 20 times a second, 50 times a second. This is also useful in diseases like cancer, because some cancers are being associated with upregulated expression of specific genes. What else do we need to? There are some sources of error, RNA degradation. We talk about some of this, and some best practices, use of replicates and other things. These are easy to read. We can move to the next slide. Thank you. Proteomics. So people have this saying that proteomics is where the cell stops making promises and starts showing receipt. What they mean is, just because there is a gene and that gene is being expressed does not necessarily mean that the cell would translate the RNA transcript into final protein levels. So for instance, we have a gene. That gene is transcribed into RNA. That RNA may be degraded by the cell. It may not be translated into a protein. I think I read somewhere that the amount of RNA that is actually translated into protein is around 5 to 10%, compared to 85 to 90% of the exome that is translated into messenger RNA, RNA rather. Okay, so how do we do protein analysis? We also extract it as before, like in transcriptomics and what have you, and then we check the quality. Sometimes you do SDS patch, sodium dilucyl sulfate, bleacromyl gel electrophoresis. It will tell you the quality of the protein that you have, and then if you confirm the quality, you can go ahead and do cleanup of the extracted protein to increase the quality, so that you can have what is called a protein isolate, something like that. And then we move to the next stage, which is spectrometrics, that is like liquid chromatography, mass spectrometry, or MALDI. MALDI is metric-assisted laser desorption ionization. Sometimes they combine it with TOF, time of flight mass spectrometry. It will tell you the mass to charge ratio of the protein, and based on that, you can putatively identify the protein. And then we move to the bioinformatics workflow. You identify the identity of the protein based on the amino acid sequences that we have, and then you can visualize the protein, including its 3D structure. And we also have databases that you deposit your protein sequence in, like PRIDE is one database that they usually deposit proteins in. And you can also visualize protein-protein interactions. I think we talked about this before. Another thing is interactomics. I also talked about this before, protein-protein interactions. And proteins, sometimes they don't function immediately after they are synthesized. This is usually the thing with proteins. They need cofactors, or sometimes they need PTM. PTM is post-translational modification. After the cell has synthesized the protein, it would not be immediately utilized. It would need to undergo some transformations before it can finally become useful in the cell for the process it's meant for. Next slide, please. Thank you. So these are some sample hypothetical data sets. The circular thing is the whole genome sequencing map. The whole genome sequencing map, what it's trying to do is, it is trying to map the entire genome in a circular fashion. The entirety of the genome would include the CDS. CDS means coding sequences, the transfer RNA, the ribosomal RNA, and it also tell you the GC content. GC content is amount of guanine and cytosine bases in the DNA. Organisms are usually classified based on their GC content into those having high GC and low GC, and this would influence the properties of the organism. On the left is a BLAST table that is comparing the percentage identity of a deposited sequence with other sequences in NCBI. Sometimes they use the table, but the more acceptable form is the phylogenetic tree, which would show you graphically how your isolate fits among other isolates. At the bottom, those green lines we are seeing is the query cover, the percentage of your sequence that match other sequences. And this is actually the percentage identity that we see. They say 99% accuracy, your isolate is Pseudomonas, for instance, or Escherichia, or Bacillus, things like that. What else? This stacked bar chart that we are seeing is showing us taxonomic composition of a community. They would say the percentage, it's usually adding up to 100 always. the microbial community is 100%, out of which 40 are this genus, 20 are this genus, 10 are this genus. So if you compare them side by side, you can easily see the microorganisms that are having a higher percentage, that are dominating, if you like, the community compared to those that are usually found in small amounts. Alpha diversity, we've explained this before. Beta diversity. Okay, so beta diversity is trying to show us, it's an improvement of the alpha diversity. It's trying to show us how equally distributed the organisms are. So for instance, if you have 4 organisms, and they said the total number of species, or not species, the total number of organisms is 12, and you have 4 species. So if they are equally distributed, then you'd expect each species to be having 3 members. 3 by 4 would give you 12. But if you have total number of organisms is 12, total number of species is 4, and then you have one species having 8 members, another having 1, you say that the beta diversity is low. So the beta diversity is like a fair share of the microbial community being equally distributed. Next slide. Okay, transcriptomics data. This is the table I'm telling you about before, that we have various genes, and in the control, how are the genes expressed in sample 1, sample 2, sample 3. This is important in medical context. So for example, you are comparing a patient with a particular disease to a healthy control. Then you can compare the gene expression in the patient to that of the healthy control, and then you can see the genes that are up expressed or down expressed in form of the RNA. And you can change this into a heat map because it will be easy to visualize. Usually you have to do Rho-Z transformation. It's a simple statistic. You can do it using R, even Excel. So basically what is showing is the genes that are colored blue or light, they are negatively expressed or downregulated. The ones that are colored red are positively expressed. You can also use PCA, principal components analysis, to visualize the groupings. What else we have? We have differential gene expression, log fold change. We've described this before. Okay, so if you look at the MA plot and the log fold plot, the blue ones negatively expressed, the red ones positively expressed, but you see some gray ones at the center. These are the ones that are neutral. They are neither upregulated or downregulated. So it means they are not affected by the stress or the disease or what it is. What else do we have? Okay, gene expression network. You know, genes have this effect where if you affect one, it usually affects other genes. So you can visualize this in form of a network diagram using advanced statistics. Next slide. Thank you. Proteomic data sets. We have the basic outcome from liquid chromatography, which is the mass to charge ratio, which should give you an idea of the amount of protein. And you can convert this into a table, which is easier to understand. How does the protein level change between the expression of the protein level change between a control and a treatment? You can also visualize it using the graphical method we just finished explaining after you do Rho-Z transformation. And you can also view protein-protein interactions. And you can have a bar chart. It's often simpler. You have the list of proteins and their expression levels compared to each other. So you can pinpoint biomarkers. Okay, these specific proteins are associated with disease or stress or things like that. Next slide, please. Okay, so how do we integrate all of this data? For instance, you can have genomic data of a microbial community, one level, transcriptomic data showing which genes are being expressed in high amount by the community, 2. And then number 3, proteomics data, which proteins are translated from the transcriptomic data. And then metabolomics, what are the metabolites that are being produced? So for instance, you are studying stress tolerance in plants, and you have the hypothesis that the plant will be producing amino acids like proline to help it cope with drought stress. If you are measuring metabolomics, you can compare the amount of proline that is produced in the stress treatment compared to the control. And then if you see a higher expression, you can say, okay, probably proline is a biomarker that is associated with stress tolerance in plants because it can help them regulate the fluidity of their membrane, something along these lines. So example of integrated output include gene copy number, mRNA copy number, or principal component analysis, and pathway. Because I told you if we have RNA transcripts, we can translate that into microbial pathways. Next slide, please. Okay, how do we apply multi-omics and biotechnology? We can apply it in many fields. Many fields, for instance, in healthcare, and precision medicine, drug discovery, vaccine development, in food science as well, functional foods, fermentation, impact of probiotics on the gut microbiome, alternative protein sources from plants. We can apply them in industrial biotechnology, production of drugs, production of bioplastic, public health in genomics of pathogens. We saw that during SARS-CoV, COVID-19, how the gene sequences are being rapidly shared all over the world. In agriculture, which is my area, plant microbe interactions, biofertilizers, stress tolerance, plant breeding. In environmental biotechnology, bioremediation, wastewater pathogen monitoring. In animal biotechnology, increasing food efficiency. Likewise, in biodiversity conservation. So any field that you take, remember one of the things I said earlier, these three things, I consider them the future of life sciences research: multi-omics, biotechnology, and bioinformatics, because you need bioinformatics to make sense of the multi-omics and biotechnology data. Next slide. Thank you. So this session has been trying to instill in us these philosophies. Number one, OMS are the entities, while omics is the study of the entities and the tools we use to study them. Number two, central dogma: genomics, the gene; transcriptomics, the RNA; proteomics, the protein. Number three, every omics is telling us a different side of the study, of the story, so we need to integrate them before we can have the final picture. Number four, we need to be careful with quality control to avoid contamination. Number five, pipelines require discipline. You have to be disciplined in form of replication, in form of even trial and error. Sometimes anybody who has done PCR would know what I'm talking about. Sometimes you have chimeras, you have wrong sequences. It can be frustrating. But actually, when you do it successfully and you have the data set, which is point number 6, then you will feel happy, I assure you. Because sometimes by just looking at the data that you obtain, you can begin to hypothesize and have insights. Okay, this gene is affecting this. So multi-omics gives us a deeper insight into biotechnology. Next slide. Okay, so we have this evaluation task. This evaluation task is telling us about a patient that was brought to the hospital. He's a 70-year-old patient. So they collected samples from this patient and one control, and they did all the omics analysis we talked about: meta-transcriptomics, meta-genomics, meta-proteomics. So they see that in the patient, the amount of the Clostridioides difficile, usually called Clostridium difficile, the amount in the control, relative abundance of the taxa after metagenomics, is around 24% in the control. But in the patient, it's around 30%, which means it has increased in the patient compared to the other external groups. They also do qPCR for toxin genes, and they find out that it's positive in the patient, but negative in the control. They also do differential gene expression, which is the metatranscriptomics, and they found out that the expression of the toxin gene in the patient is higher than in the control. And then they do proteomics, and they see that the toxin protein has been increased in the patient compared to the control. So what do we want to evaluate here in this task is, how do we compare between the metagenomic signatures between the patient and the control? And how do we link this to the potential development of disease in the patient? And do we think this disease is caused by a particular bacterium, or we have some information that is still missing? And what is the next step that we are going to take? So if this is a classroom, I would give time for people to cogitate and think it out. I think I'll usually give the pointers, not the whole answers. The pointers here are, in the metagenomic dataset, we see an increase in the Clostridium difficile expression, which is telling you that this patient is likely having antibiotic-associated diarrhea. Clostridium difficile is usually the causative agent of AAD. AAD means antibiotic-associated diarrhea. And based on the qPCR and transcriptomics and proteomics, we can see that there is an increased level of toxin secretion, probably from the Clostridium, which is perforating holes in the intestinal membrane and leading to the leaking of the electrolyte, which is basically the diarrhea. So we can suggest, based on this data set, that, okay, probably this patient is suffering from antibiotic-associated diarrhea. So what was the antibiotic that was being given to him before? Let's remove it, substitute it with another antibiotic that can take care of the Clostridium difficile that is being elaborately expressed and having a higher taxonomic abundance. Next slide. Okay, these are some selected references. I have one or two of my works that I did about multi-omics, even though they are just elementary. And then we have general references that we can read to learn further. I apologize for the lecture being a bit rushed. It is impossible to cover multi-omics in biotechnology in just a single lecture. It's impossible. It requires a series of lectures. But the bottom line is I'm trying to shed light on the key aspects, and then students can explore further at their own leisure. I have this quote that I really like from Isaac Newton. He said, "If I have seen further than others, it is by standing up on the shoulders of giants." If I was able to do anything good today, it's not from me. It is the efforts of my teachers who have been teaching me, and all the people that have been publishing wonderful papers that I have the privilege of reading, and VitaNova International Alliance for Sciences for finding me worthy of delivering this lecture. I utilize some artificial intelligence tools in the design of the slides. I have attached them below. And the first slide is gotten from Slides Carnival. Thank you, everybody. I'm really grateful.
Thank you very much, Mr. Yahya Yunusa Riko, for such an insightful and inspiring session. We will wait for a few moments so that we can reach out to the students who are having doubts or questions or confusions, and the participants can write in the chat box. In the meantime, I'm sharing the attendance link so that the participants can fill the form and mark their attendance. Remember, any kind of excuses won't get considered if you do not fill the form. So kindly fill the form as soon as possible and kindly share your confusions or questions with us. So the participants are now open for Q&A session. Okay, I can see the praises and the compliments here more than the questions. So, do you want to share any kind of experience that you have with us for this session? Kindly, the floor is yours.
Okay, thank you very much, man. My experience is, I think, in around 2019, after I finished my bachelor's degree. So my supervisor, Associate Professor Zubair Umar Dharma, he said to me, we are going to write a review. I was thinking of writing a general review, narrative review. These are the reviews, topics that have been covered before. They find this result. He said to me, no, go and talk about something that has to do with molecular biology. And that advice changed my life. It's actually the first time I looked in depth into the multi-omics, and then we wrote a review paper about it. I attached it in the reference. I think it's the first reference under my works. So after I read that paper, I understood that the world is gradually moving from traditional approaches to multi-omics. And then later during my master's degree, I focused on the phenomics and metabolomics. I studied the rhizospheric microbiome of the sorghum bicolor. It's a variety that is found in Nigeria that has a very high phytoremediation ability. And then now at my PhD level, this is where I am really exposed to all of the metagenomics, metatranscriptomics, metaproteomics. Even though I'm just starting, I still have a lot to learn. So my advice for students would be we should always have curiosity. Should always have curiosity. If you read a paper in any high impact journal and you see they use this metagenomics, multi-omics, instead of reading the paper, I'm like, this is really nice. You should be thinking, how can I apply these techniques in my own research? What are the insights? What are the ideas that I can generate from? Because basically, to me, multi-omics is a gold mine. It's a gold mine. There are a lot of things that can be discovered. Like they say, we have not even started yet. We have not even scratched the surface of, because look at the complexity of the genome. The genome of even a bacterium is highly complex. If you have the data set they usually give after they do whole genome sequencing, you would be surprised at the amount of information that is contained in the data set. So we should have curiosity and we should also collaborate. I know some of us in the areas that we are in, we cannot do all of this analysis because we lack the infrastructure. So what I would advise us to do is to have collaborations. Somebody is in a different part of the world and he has the tools and techniques and he needs someone to collaborate with. By even a simple review, sometimes you can do a review. You gather all of the studies that some people have done and then you produce a review and you can gain insights. For my master's, I did a review on whole genome sequencing. It was published as a book chapter, also included in the my work section. And that is where I learned some more about whole genome sequencing. So the importance of this, remember the three objectives of the session. You need to have the foundational knowledge first before you can apply this into your own experiment, and then you use this to understand the data that you're going to get. So my advice, stay curious. If you see somebody do a good work, think, okay, I can do this as well. Collaborate, start with reviews so that when you are in an environment that you can finally explore it, you can explore it. And I believe you can do it. Thank you very much.
It's truly feeling honored to know about your experience, know about your journey. Thank you so much for being our guest and being our speaker, training our students from all over the world. Your discussion on omics technologies in modern biotechnology has given us a valuable understanding of one of the most rapidly advancing areas of life sciences. We sincerely appreciate your time, your knowledge, your dedication in making today's session so informative. And I would also like to thank all of our participants for joining us today. We hope to see you again in our upcoming sessions as we continue exploring the latest advancements in molecular biology. Thank you, everyone. I love it, and I have a wonderful evening. And thank you as well as Mr. Yahya Yunusa Riko. See you in a future collaboration as well.
Thank you. I'm really grateful and honored. I appreciate this opportunity.
That's really great. I love it.
Thank you.